So let's get started with the general idea why we are talking about this. I think we have already shown this in former lectures. We got interested in knowledge graphs because we found out that there's much potential in using graphs for organizing knowledge. On the one hand, we have the data side. So if you have a big database, you can annotate the data in a classic way with tables and metadata files and so on, but you can also organize them with a knowledge graph, so in a graph-like hierarchy, which makes data much more accessible. And this is, for example, the case in the eBrains database of the Human Brain Project, but also for many other rising databases that use this technology. And on the other hand, we also have knowledge graphs about biological knowledge, about pathways, for example, developed by the Fraunhofer Institute of Scientific Computing. I think we have shown some about neurodegenerative diseases. We are working on one about psychiatric diseases. So they explain the relationship between, for example, proteins, genes, and the processes between them. And on the other hand, we also are developing an ontology that organizes knowledge about the vertebral brain in a graph. So an ontology is always a bit like a graph. So it contains the knowledge about what can be modeled in TVB and what is the relationship in between all these parameters. And what has this now to do with anatomy? The problem was that the knowledge about pathways is not anatomically represented. So we know the relationship between genes and proteins, but we don't know where these processes actually take place. And this was a gap of knowledge which we wanted to fill with our software tool TVBase, the Virtual Brain Adapter of Semantics. And how does it work? We are using an existing tool from, again, from the Fraunhofer Institute called Skyview, which is just a literature database that contains all the papers that are also contained in PubMed. So it's more than 30 million scientific papers. And in these articles, the words are annotated. So you can search for something, for example, for protein, a gene, but also for free text searches or for mesh terms. And then you get a list of the results. And the extra feature is that you can extract statistics about these results, because all these papers were gone through text mining, and therefore we know, for example, the genes that are mentioned in there, but also the anatomical terms in the Uberon ontology. And they describe, for example, the primary visual cortex, and also describe in an ontology what is the relationship in between these anatomical terms. And therefore you can extract statistics of how often a particular term is mentioned in a search query. But what was not integrated and is now a bit, now it works again, the video. What was not existing is a relationship of these semantic anatomical representation and the three-dimensional anatomical representation. And therefore we manually assigned these Uberon terms, about 1,000 Uberon terms, to the 3D anatomy of the brain. We used the FS average template for the Glasser parcellation, and manually assigned it in a detailed anatomical curation. For example, where exactly the borders of the temporal lobe are, but also of much more smaller and particular regions of the brain. And therefore, our approach is that we extract statistics, and therefore it's kind of a meta-analysis, semantic meta-analysis of scientific literature, and we transform these statistics onto the 3D anatomy of the brain, and therefore you can build brain maps that map semantic knowledge. This is actually what TVBase does, and as you have now heard a bit, it has had the core of, or one of the core features is the transformation from the semantic Uberon space to the 3D anatomy. And this is the point where you need the anatomical knowledge to do this curation and to do it as precise as possible. And therefore we give this talk. And now I will hand over to Konstantin.
Yeah, thanks. So anatomy can sometimes be a bit dull and boring, so we decided that we are doing multiple changes in between to keep you all awake. And as Leon said, this is not a substitute for a proper anatomical lecture. It's just some fundamentals, some basic knowledge that we wanted to share, and that was also necessary for this translation of the semantic Uberon terms to the Glasser parcellation. Yes, and that is okay. So first of all, how did we achieve this transformation? We did this by first extracting all anatomical Uberon terms that are annotated in SkyView, which are about later pointer, which are more than 10,000. And these, for each Uberon term, we then try to find the best fitting description of its area in the Glasser parcellation. For this, obviously, we first refer to the Glasser works itself, which actually had a very detailed neuroanatomical supplementary where a lot of the terms were described directly, which region in the Glasser parcellation they refer to. If the term was not mentioned, we refer to the very detailed neurosurgical works by Baker, who in, I think, 8 publications described each Glasser area in much detail and the exact anatomical location. If we didn't find a description there, we refer to the Desikan and Destrieux parcellations. And these, if we found a term that fitted the description of this parcellation, we could actually use automatic vertex matching, because these are already parcellated areas. And if we didn't find a description there, we refer to alternative literature, excluding the term if there was nothing found. And for each of these Uberon terms, we then try to find a Glasser area as either an exact match, if the Uberon term referred to the same area as the Glasser parcel, for example, here the primary visual cortex, or a subclass if the Uberon term referred to only part of a Glasser area, for example, here the corner munus of the hippocampus as a subclass of the whole hippocampus, or as a superclass if the Uberon term referred to more than one Glasser area, for example, here the temporal lobe that refers to around 32 Glasser areas. And in this process, we actually created a very detailed anatomical documentation where we documented and discussed each difficult assignment. And overall, we assigned more than 1,000 Uberon terms to different Glasser areas and also to subcortical fissure segmentation areas. And yes, we just wanted to share some of the knowledge we gained there, or some of the knowledge that we reignited from our medicine studies. And maybe to start in a very, so before we go into the details about the cortical and subcortical structures, we wanted to give a very broad overview of the nervous system as a whole, because the nervous system is not just, so to say, the brain, or not just cortical and subcortical structures, but it's also the cell components of it, so the functional main component, the neurons, and the the other main component, the glia cells, both make about, in an adult brain, are about 80 billion cells. And the glia cells is a superclass of different cell types. So one cell type is depicted here, the astrocytes, which actually are kind of the connective tissue of the central nervous system and have this kind of star-like structure, as you can see here. That's also where the name is coming from, and which are important for regeneration, also nutrition of neurons and other glia cells, and also from the blood-brain barrier. And the other type of glia cells are the oligodendrocytes, which form the myelination sheath around the axons in the central nervous system. That kind of acts as a electrical insulation barrier. Then ependymal cells that line the spinal cord and the ventricular system of the brain, and the microglia, which are actually involved in the neuroimmunology of the brain, and are kind of specialized macrophages that also are kind of, so they fight, find any bacterial or viral agents, but also are kind of clearing out the debris, also kind of the trash mans. And in the nervous system then as a whole can further be differentiated in the central nervous system that is constituted of the brain and the spinal cord, and the peripheral or peripheral nervous system. So the central nervous system I will refer to as CNS, and the peripheral nervous system as the PNS. And the glia cells in the peripheral nervous system are quite different than the ones from the central nervous system. All the cells I just listed in the central nervous system don't exist in the peripheral nervous system, or the PNS, but actually only the so-called Schwann cells, which are also important for the myelination of the axons. And the peripheral nervous system actually contains a lot more nerve fibers than nerve bodies, and a lot more nerve fibers than the central nervous system. And one more important differentiation for the central nervous system is the differentiation between gray matter and the white matter. And the gray matter usually contains a lot of cell bodies of the nerve system. So this is cell bodies of the neurons and also cell bodies of different glia cells. And the white matter actually has only very few cell bodies and a lot of myelinated axons. And this is also the reason why it is called white matter, because this myelin contains a lot of fat, which then in histologic slices, or if you look at the live brain, looks a bit white or whitish, at least whiter than the gray matter. And the gray matter is usually grouped into distinct nuclei, especially in the spinal cord, and also in the deep basal ganglia, which is then surrounded by white matter, except for the cortex, where the gray matter actually surrounds the white matter inside. Okay, so, and maybe one more point: the differentiation between the glia cells of the PNS is not arbitrary. It actually derives from the embryogenesis, and this is because the glia cells of the peripheral nervous system actually derive from the neural crest, while the peripheral the central nervous system glia cells and also neurons derive from the neural tube. And this knowledge about embryogenesis is a bit hard to grasp, and is also always very hard to learn I found, but it's actually very helpful when differentiating between different parts of the brain and really understanding why these parts are different in function, but also different in location, and why different parts are really interconnected, for example. And if we look at the embryogenic brain, at week 4, this neural tube that makes up the central nervous system, so the neurons into the central glia, differentiates in 3 distinct neural vesicles, which are called the rhombencephalon, or the hindbrain, the mesencephalon, or the midbrain, and the prosencephalon, the forebrain. So 3 vesicles, which then at week 5 differentiate in 5 distinct vesicles, which are then called already this, or the prosencephalon differentiates in the telencephalon and the diencephalon. The mesencephalon stays the same. The rhombencephalon differentiates in the metencephalon and the mielencephalon. And also the spinal cord differentiates at this time. And maybe if you want a trick to memorize these 3, the order of these 3, it's so mesencephalon is alphabetically before the metencephalon, which is alphabetically before so the Y of the mielencephalon. But it's not that important. But also what you see here, one thing that happens around this time is that the telencephalon actually stretches out into two directions, actually forms a kind of T. And these 2 parts that stretch horizontally will then form into the 2 distinct hemispheres. And another thing that happens on this at this time is that the telencephalon, while it kind of stretches out horizontally, it also tilts in comparison to the diencephalon. This is the reason why these axes that we see here are also tilted in the brain. So these neuroanatomical axes, usually, if you describe the body, you always describe it out of the view of the patient. So, and usually the backside of a patient is always dorsal, the front side ventral, downwards caudal, and upwards cranial, or in the case of the central nervous system, rostral. But due to this embryogenetic tilt of the axes, now the actual for the telencephalon, the axes go ventral, actually in the caudal direction, rostral in the ventral direction, dorsal to the top direction, and caudal to the back direction. So if you have neuroanatomic literature and are very confused about the orientation, this is why they refer to these completely different axis description. And this blue axis, this tilted axis, is called the Farrell axis, and this non-tilted normal axis is called the Minot axis. Okay, these neural vesicles then differentiate further into the telencephalon, which is basically what's called the cerebrum, which is usually what you mean when you talk about the brain. The diencephalon, which is located here, and which actually extend. You can see this in this abstract figure. Extends up until the retina. So the retina, and therefore the eye, is connected directly to the central nervous system, to the diencephalon. Below this, we have the mesencephalon, the midbrain. The metencephalon then differentiates into the pons that lies before the cerebellum. And we will see that these 2 structures are actually quite interconnected, and this stems from this neural embryogenesis. Below that, the medulla oblongata that then further extends into the spinal cord. Okay. The cortex, so the cortical gray matter that we see here on this lateral view of the left side, has a lot of So it has a very complex folding pattern. And this folding is done so that we can actually fit more of this cortical gray matter inside our limited volume of the skull. And these folding patterns have convolutions and also these bulges, these gyri that are separated by sulci or one sulcus, and which is a more surface groove, or if it's a bigger groove, a fissure. And by this morphometric structures, so the sulci and fissures, we can actually differentiate between different lobes. This is not the only differentiation. The most important differentiation is actually by function, but it helps in differentiating this. And we can see here's the frontal lobe that is separated by the central sulcus from the parietal lobe. And then the temporal lobe that is separated from the frontal lobe by the lateral sulcus. And now this insula you usually don't see if you look from lateral on the brain, because it's completely covered by the operculum. And inside this lateral sulcus, underneath the operculum, the insula lies. And actually with this folding pattern, or so two more facts, so the cortical gray matter, more than 60% of it lie within these deep grooves, so within these sulci and fissures. And it's possible to achieve a area of around 1800 square centimeters with this folding structure. And here again, you see these different sulci and these cortical gray matter that lies within or underneath the surface. And here another important fissure, the parietal occipital fissure, that separates the parietal lobe and the occipital lobe. Okay.
Okay, so I will continue now, and because we also want to have to make this talk a bit interactive, we can provide you one tool of our group, which is an interactive atlas that allows to explore the anatomy of the brain. So there will come a version soon where you can also explore pathways and transmitters and other things, but there is already a public one where you can have a look to the lobes of the brain, and their function and so on are explained. And here we use it to show you a bit what will come now. So we will now go into the detail of the lobes of the cortex. And this is also how it looks in this atlas. And on the anterior parts, on the front side, up to the eyes, we see the frontal lobe. And then on the lateral parts of the brain, a bit posterior are the parietal lobes, and the really posterior parts are the occipital lobe, which is called also the occiput, the back part of the head. Now the video lags a bit. This is also where the visual system lies, and then the temporal lobes are around the ears. So this is the temporal part, and then we also have a lot inside, for example, the limbic system, gyrus cinguli, and so on in the inner part of the brain. The insula, as Konstantin already explained, inside of the cortex, therefore it's also called insula. The cerebellum below the occipital lobe, and here from downside we see also the brainstem and all the other nuclei like the hippocampus, the amygdala, the basal ganglia, which are inside, so you can only see it if you make the rest of the brain transparent. So now we will start with the frontal lobe. It's one of the biggest parts of the cortex, and probably the most well-known function is that it's the lobe where the primary motor system lies. So Brodmann area 4 or M1 is the primary motor cortex, and the gyrus praecentralis lies in the frontal lobe, in the beginning of the frontal lobe. And there you have the projecting pyramidal cells in the somatotopic organization. So there are all the muscles represented. So this is the part of the brain where all the movements, the intended movements come out by projection down from the primary motor cortex through some regulation in the cerebellum down to the spine, and then through the nerves directly to the muscles, where you see then the electrical activation and the muscle contracts. This is the main thing for the primary motor cortex. But as the movements that human people do are more complex than just activating one muscle, we need also some more motorical areas, and this is the premotor cortex, which is involved in movement planning. And we also see, for example, in fMRI studies, activations of the premotor cortex and also the supplementary motor cortex. It's even a bit more anterior when you are planning a movement. So when you just imagine to do a movement with your right arm, you can see this, because then the planning regions in the frontal lobe are already activated. And if you are really doing the movement, then also not only the planning regions, but also the actual motor regions are activated, and then a signal goes down to the muscles. And this contains also the frontal eye field, which is a very specific part of the motor cortex, because it controls the voluntary movements of the eyes. So I will not go into detail about different kinds of eye movements, but interestingly, you can try it out. It's not possible to move the eyes slowly without an object that you are following. So if you follow an object, for example, a finger, you can do this slowly. But if not, you can only do fast eye movements. We call this saccades. So it goes from one target to the other one. And these are voluntary eye movements, and they are controlled by the frontal eye fields, which lies there also in the area of the supplementary motor areas. And what is also a part of this is Broca's area, which is responsible for speech production. And this is all, if people have a lesion in Broca area, the so-called Broca aphasia, then they are not able to produce the movements that are necessary to produce speech. So it's not a paresis of the speech muscles, but it's a problem in executing this movement program. And therefore, this Broca aphasia is also often called this so-called motoric aphasia. And the second important part of the frontal lobe is the prefrontal cortex, which sounds a bit counterintuitive, but this is because from the clinical neurological view, the frontal cortex is very strongly assigned to this motor areas, because, for example, these are the main problems in stroke patients and so on. And therefore, the term prefrontal cortex was invented for the anterior part of the frontal lobe, which contains completely different functions. So especially for the supplementary motor areas, the transitions are a bit fluid. But normally, if you refer to the prefrontal cortex, you mean this part of the frontal cortex where abstract thinking and personality are lying. This is probably something you all know. And a lot of different functions are in there, also association areas. But I think important to know is that there are you can still subdivide the prefrontal cortex into three big area groups. One is the dorsolateral prefrontal cortex, which is important for planning. So it's not planning movements, but a bit more on an abstract level, on a meta level, planning, sorry, maybe we can go back, planning general things. And this is what we call executive function. And if people have an impairment there, we call this in neurology a dysexecutive syndrome. And this is really that you can see in these patients that they are not able to organize themselves to plan an action. So if they want, so they want something, for example, they go to a shop to buy a sandwich, and then it's difficult to really get started for them to ask the person in the shop that they want the sandwich and so on. So this is what we call a dysexecutive syndrome. So if you have a strong dysexecutive syndrome, it's a really strong impairment of daily living. And then we have something a bit similar on the medial part of the frontal lobe, near the cingulum, in the anterior cingulum. We see something. There is more we call this psychomotorical activation. So if you have a problem there, people are getting slower in their thinking, but also slower in their movements and in their actions. And with a very strong lesion, they also can get akinetic, so they are not moving anymore. And this is again, this is what you see, that people are not moving physically, but it's also something of the mind, that the mind is not moving anymore. So this is also these people are building a apathy phenotype, and this is, for example, the case in particular forms of frontotemporal dementia, also, or in many neurodegenerative diseases, that in the end people end in akinetic mutism, so they are not moving and not speaking. And this is a very this is the syndrome of the medial prefrontal cortex. And then the most prominent syndrome of the prefrontal cortex is the disinhibition syndrome in these orbitofrontal and orbicular areas. So the inner lower part of the prefrontal cortex, this is where the social control lies. So these are things that normally, these areas normally control our behavior, so that particular actions do not happen because they are socially not as accepted, but also because from an ethical perspective, we don't want to do them. It can be, for example, also to avoid violence and all these things, but also, for example, to not eat every cake that you are seeing. And if you have a problem there, or a lesion, then all these mechanisms can go away, and then people often get aggressive, often very angry, can't control themselves, follow every instinct, and so on. And the most well-known patient was Phineas Gage, and a very early patient here who had a lesion caused by a metal, a big metal piece that in an accident lesioned his orbitofrontal cortex, and this led to very low deficits in the general neurological function. Therefore, people found this case very interesting, but the personality was completely changed, and he got very angry and very, very special, and the whole personality was changed, and therefore this was how this function of the prefrontal cortex lower part was found out. Okay, so now we will continue with the parietal lobe.
Yeah, the parietal lobe lies just behind the frontal lobe, separated by the central sulcus, and is most famous for its role in somatosensory information well, in receiving somatosensory information. And there we differentiate between the primary somatosensory cortex as one that lies on the gyrus or gyrus postcentralis, or postcentral gyrus, sorry, it's a very German, Latin, English, and which is intensely connected to the secondary somatosensory cortex. And this primary somatosensory cortex receives information from so about different qualities. So about the first one is called the protopathic sensory information, which is actually refers to pain and temperature sensation, and also the epicritical sensory information, which refers to the sensation of touch, so a very fine-grained sensation, also the conscious perception of where your body is in space. So knowing that your muscles are, or knowing how your joints stand to each other, knowing where your arms are at the moment, this is all because of this epicritical so-called proprioception. And this information is relayed usually from receptors inside the skin for the epicritical information, or inside the muscle cells or the joints, and then travels through the spine, or in this case of facial sensory information through the medulla oblongata up to the thalamus in the diencephalon, and then which is on the contralateral side, and then goes up to the primary sensory cortex. And here, as in the primary motor cortex, we have this somatotopic organization, and we see here that some parts of the body have a much larger representation on this primary sensory cortex. And this you can also try out. So for example, if you close your eyes and then just, ideally by someone else, but you can also try it yourself, just try to discriminate between 2 points on your hand in comparison to 2 points on your lower back. You will see that it's much harder to differentiate the lower back because it has such a smaller representation on the cortical surface here. And another very important functional feature, which is actually true for all sensory cortices, this primary somatosensory cortex, as well as the primary auditory cortex or the primary visual cortex, receives the information, but is not able to interpret this information. For this, they need further association areas or further kind of interpretatory cortical areas, for example, this secondary somatosensory cortex. And here, the understanding of what we are touching or what we're hearing is actually developing. And this is also true if you have a patient with a lesion or with this secondary somatosensory cortex is not working. He will develop something called tactile agnosia. So agnosia from do not recognize. And if you give this patient something inside his hand, for example, a pen or a ball or whatever, he will feel the sensation, but he's unable to interpret what he's touching. So even if he feels it, he can't tell if it's a ball, if it's a pen, if it's whatsoever. And this is also true for the other sensory cortices. And so as I said, the sensory fibers come from the contralateral side, and there's also a syndrome called neglect that is happening if you have a lesion on your non-dominant side. So that is for most people the right hemisphere, because most people are right-handed, and the contralateral side is then the dominant side. And these patients neglect their whole contralateral body, so to say. So they do not recognize their contralateral body half, especially the visual part, but also do not see it as pathological. So it just doesn't exist for them. and some more syndromes that are associated with lesions in the parietal lobe. One is maybe we start with this apraxia. As I said, the neglect is usually happening when you have a lesion on the non-dominant side. The apraxia is, on the other hand, is happening if you have a lesion on the parietal lobe, especially in these posterior association areas on the dominant side. And this is then a lot more movement orientated. So these patients are not recognizing their contralateral body part, but are actually incapable of reproducing learned movement. So this can have different forms, but for example, they cannot carry out a motor command, or they cannot plan any specific movements. And this you can also, so these are TV-based maps, this interconnection to the motor cortex, you can really nicely see here. And this is just to show how these different lobes, different cortices are highly interconnected and need the association information or the different sensory information from different parts of the brain to actually function properly. Another syndrome is the Gerstmann syndrome, which is also usually caused by stroke, tumor, or hemorrhages in this area, and especially in two different gyri. So the first one is the gyrus or supramarginal side gyrus, and the angular gyrus that you can see here on this TV-based map. And these patients typically also have problems with functions that need the integration of different modalities. For example, they cannot drive, so that's called agraphy, because there you need the integration of understanding what you're writing. So the kind of that's also related to temporal areas, the writing, which is related to motor areas, and then this integration in the parietal lobe, that it cannot happen. They are also unable to read usually, and also have a difficulty, which is called a toe finger agnosia, difficulty to name their own fingers and toes. And this is all happening without any intellectual deficiency. So the intellect is normal, they have just these very specific loss of integrative function, for which the parietal lobe is so important. And this dyscalculia refers to the parietal lobe also as the so-called mathematical cortex, because it's highly relevant for being able to do math. And the next lobe we were going to talk about is the occipital lobe.
Yes, and as I shortly already mentioned in the beginning, the occipital lobe is the main lobe for the visual system. I found it always quite easy to recognize this if you imagine the visual system in the brain a bit like the projection in a cinema. So the projector are the eyes, and it's projecting the picture onto the screen, which is the occipital lobe on the other side of the brain. Actually, also the fibers that are going from the eyes to the occipital lobe are also called radiation. So the radiation of vision. And I will not talk about what happens before the occipital lobe, because it's maybe some of you know the crossing of visual fibers is quite complicated, but in the end we get a whole representation of the picture that is projected on the retinae, also on the occipital lobe. So it's not in the same order, because, for example, the left parts of each retina are both on the left side of the brain. But in principle, the picture is really one-to-one projected to the brain. So you can really see the patterns. So this is what the primary visual cortex does. So which you can see in the sulcus calcarinus. So you can see this as a structure on the occipital lobe. It's really the most posterior part of the brain, and there it's really, as I described, a bit like pixels. So you just see the colors and the lightness of each cell on the retina, a bit like a camera also does. But this is still not sufficient for our understanding of vision, and therefore you need also secondary visual areas, and these are these peristriate areas. So the more far from the primary visual cortex they go, the more abstract is the processing of the picture. And it starts with basic image processing, like, for example, decoding the color and the form, the contours. So to make from just the pixels patterns that can be recognized later on. Interestingly, this is very similar how, for example, also machine learning for mimicking visual perception works. And later on, these patterns, so the processed picture with the contours and borders, in particular borders between lighter and darker areas are important here. These are then interpreted because then, for example, patterns are recognized or faces are recognized. If there are faces, then the distances between the eyes and the nose and so on are automatically analyzed. This is important because this is how we recognize other people's face, that we know these people already. And this goes into two directions from the primary visual cortex, which is actually which are called the dorsal and the ventral stream. And to explain them very easily, the dorsal stream is the so-called where stream, and the ventral stream is the what stream. So first, why are they called stream? Because it's really, as I said, the unprocessed image is in the posterior part, and then it goes further and further, and the processing goes gets more complicated the further you go. Therefore, it's this information stream, and it goes into two directions. And the where stream goes to the parietal lobe and the occipitoparietal areas where the spatial orientation is represented. So this is important to analyze the spatial information in a visual picture. And this is also, for example, therefore this is a very important input for our spatial navigation at all, because our spatial navigation works mainly based on the visual system. So the input from the eyes and then from the occipital lobe gets into these areas that say all the time to our brain where we are physically. And it's necessary to get feedback from the visual system where we actually were. And the what stream is more the interpretation analysis. This is, for example, one example is the interpretation of faces. That this is the last point here in the most lower part of the temporal lobe. So the gyrus fusiformis is well known for face perception. And if you have, for example, a lesion there, you get a symptom called prosopagnosia. So the people are not able to recognize faces, which is a very particular thing because faces are much more similar to each other as other things that we recognize. But our human brains are really tuned to find very small details that make difference between faces. But also other recognition is done in this what stream. So, for example, as I said, pixels are converted to contours, and then contours to patterns, and patterns to objects. And then these objects are compared with our memory of known objects. And then we see, okay, for example, this is a flower. and then this interpretation of a flower can be processed together with the other objects that we are currently seeing, and then we see, for example, this is a flower that is inside of a wooden frame, so it's a picture of a flower, or we just see a flower in a glass, therefore it's a real flower, and so on. So this is what the word stream does, and these are really completely distinct information flows that are both highly dependent on the visual input.
Yeah. Then the next lobe we want to talk about is we need to talk about is the temporal lobe, which has well, the main function has the main function of the temporal lobe is the receiving of the auditory information and interpretation of this information. So if we look from the lateral side here, we see this auditory system that receives its input from the ears, basically. And here on this inflated brain, it is quite easy to see. But in the real brain, this so-called gyrus or transverse temporal gyrus, also called the Heschel gyrus, it's actually hidden underneath the frontal and parietal operculum. And here lies the primary auditory cortex. And in the same way, as I said, with the somatosensory cortex, this cortex receives the information first, but is unable to interpret this information and needs further secondary auditory cortex and the auditory association cortex to actually perceive and understand what he's hearing. So if, for example, if you would stimulate this primary auditory cortex, you would know that you're hearing a sound, but you couldn't say if it's a melody or if it's just a white noise. And yeah, as Leon already mentioned, the ventral stream, the what stream, is also part of this temporal lobe. And another very important area is this, so the secondary auditory cortex, also called the Wernicke area, because it's, as I said, important for the interpretation, and also has different functions depending on the laterality. So on the dominant side, so for most people on the left hemisphere, the secondary auditory cortex is important for the rational integration. So it's important for the speech comprehension. And on the non-dominant side, this is more related to more emotional parts of sound. So, for example, understanding and music, and also developing emotions because of music. And this is then also relayed by lesions of these two parts. So if your dominant Wernicke area is impaired, you have a normal speech production because the Broca area on the frontal gyrus or the frontal cortex is unimpaired, but you are not able to comprehend what you're saying, and because you're not able to comprehend what you're saying yourself, it's also incomprehensible to people listening to you. And people with this Wernicke aphasia also often just invent new words as they are talking, which is called neologisms. Yeah. And on the other hand, if the non-dominant side, the non-dominant secondary auditory cortex is impaired, then people lose the ability to understanding and recognize music, which is also quite sad. And on the medial part of the temporal lobe, there's actually a lot of stuff going on, because this is kind of a transition zone between very different types of cortices, but also different cortices that are highly interconnected. and also the transition zone to the limbic system that Leon will talk about in a second. And here we can actually see the so-called different types of allocortices, which are actually defined as being not or different from the neocortex that we see in all other cortical parts with its typical 6-layered structure. And these allocortices parts are typically parts of the cortex that are phylogenetically very old. For example, this paleocortex, you can see it also in the name, is probably the oldest part of the cortex that we have. So it hasn't changed in millions of years. It's actually the primary olfactory cortex. So this receives direct information from the sensory fibers of in the nasal cavity, and then sends this information towards secondary olfactory cortices, for example, in the entorhinal cortex or in the orbitofrontal cortex. Another very prominent structure of this allocortex is the hippocampus, which forms the archicortex, and obviously the hippocampus is a very complex structure in itself. It's related to long-term memory, plasticity, sense of time and space, and also related to many different, especially neurodegenerative diseases like Alzheimer's. And then there are typical in-between or transition cortices. So the periallocortex, which is a transition zone between the real allocortex and the proesocortex, and the proesocortex, which is then a transition zone between the periallocortex and the esocortex or the neocortex. So it gets kind of younger still. So paleocortex the oldest, with the archicortex, then a bit younger the periallocortex, then the proesocortex, and then finally the neocortex. And these old structures are actually what also make up the limbic system. So the cingulate gyrus, this parahippocampal gyrus, and the archicortex with the piriform cortex also. Yeah, this is also why we see in our assignment here that it's kind of a layered structure, so going from inside to outside and going in different layers the younger the cortical areas become.
Okay, thank you. And I will very shortly come also to the limbic system, but just for completeness, we already told some time ago about the opercular parts of the frontal lobe. Just important to understand what this is. These are the inner parts that are inside of the lateral sulcus. So here the brain is folded into itself, and therefore the temporal lobe, but also the frontal lobe and the parietal lobe, have parts that are inside of this sulcus, and these are called the opercular regions, which also contain a lot of important functions dependent on the lobe that they belong to. And also in this area is the insula. As I said before, it's called the insula because it's really inside of this sulcus, and it's a bit apart, therefore, from the rest of the cortex. I think probably more correct would be the peninsula, so a half island. But and this is very has also very complex functions, so it's also involved in emotion processing and so on, but the most prominent function is the gustatory system, so the understanding of tastings of food, and also the integration of information through the brain gut axis. So from the bowel, we also know that the colon has its own nervous system and also a lot of receptors, and gets a lot of information, and this information also gets transmitted through the vagal nerve to the brain, and then it's integrated in the insula together with the gustatory sensory information, and therefore it's also one part of for multisensoric integration, so where all these things come together, and therefore it's also very important in latest research on understanding, for example, obesity and so on, because this is where these processes also take place. But now we come to the limbic system. Maybe just to explain why it's called the limbic system. Limbus is calling the border of something, and therefore it's named from the gyrus cinguli, which is the one gyrus that directly is placed around the corpus callosum. So, which the corpus callosum is white matter that connects the both hemispheres of the brain, and around it we have the gyrus cinguli. And as Konstantin already turned out, very old structures of the brain are lying there, and we also see a lot of things going through this circle of the limbic system. So the most prominent one is the Papez circle, involved in memory. So it's a very strong simplification, but in principle you can imagine that memories are memory consolidation starts in the hippocampus and then goes through the fornix, through this limbic lobe once again. This is a bit also very naive understanding of memory consolidation. And probably you all know the limbic system, even the word is strongly associated with emotions. So emotion processing is placed there. Therefore, it's also connected to the amygdala, which is not part of the limbic lobe, but part of the limbic system. Now you're also seeing that we are switching a bit from the structural parcellation of the brain to the network view. And this is what is not now here on this slide, but Konstantin also mentioned it, that it actually starts in the very beginning here from the olfactory system, and this is also very interesting because therefore we have a very strong connection from the olfactory system both to the hippocampus, so to memory consolidation, and also to the amygdala for emotions. And the amygdala is mainly involved in bad emotions, and this is also something that probably everyone knows, that smells are activating memories very easy, which has also to do with this very direct connection and anatomical neighborhood of these structures. And the same is that bad smells are directly leading to bad emotions in the amygdala. And interestingly, this is the only sensory information that directly goes unprocessed from the primary sensory cells, so the olfactory bulb, to a structure of the subcortex, so here the amygdala, without going through the thalamus before. So this is a very particular thing which has to do with this very old architecture of this all axis of memory, smells, and emotions. And when we continue, the slides do not continue. Yeah, we now continue with other subcortical areas. So I think we have hurry a bit.
Yeah, exactly. So yeah, I think each of these subcortical areas could fill a whole session of these 90 minutes. So if you're working with any of these, please excuse us that we don't, that we are not like going into the complexity of this structure that it actually deserves. And if you paid attention in the embryogenesis, this is going to be easier for you, obviously. And the first structure we want to talk about is the brainstem, which is actually a combination of 3 different structures, also 3 different embryogenic structures, so the midbrain, the pons, and the medulla. And this brainstem is, because of its small size and noisy surrounding, so noisy surrounding anatomical structures, and its high complexity, a area that has been very difficult to map using magnetic resonance imaging, and it's only possible to map with ultra high field MRI, and also with the classical histological slicing of this area. And in this brainstem, we have the nuclei of the cranial nerves, except that's wrong, shouldn't be there, but except the second one, except the olfactory nerve. and it's basically involved in a lot of automatic behavior, so behavior that is not consciously, that we're not consciously aware of. So at the top, the midbrain, you see a very complex structure, very difficult to comprehend. So a lot of different structures in a very tiny space. But basically here we have the nuclei of the third and fourth, third and fifth cranial nerves. The nucleus ruber, the substantia nigra, is involved in Parkinson's disease. Yeah, and this is basically the midbrain is important for arousal and alertness. Let's just sum it up like this. Then the bit caudal to this, we have the pons. And if you remember, this pons is coming from the same embryogenetic vesicle as the cerebellum, which is the metencephalon. Yeah, metencephalon. And this is why it's highly interconnected with the cerebellum. It's actually the link from the cerebellum to other parts of the brain, so the spinal cord and the cerebrum. And in this pons, we also have a couple of cranial nerve nuclei and also a lot of fibers that go upwards, sensory fibers that go upwards and motor fibers that go downwards. But again, very complicated structure, and I won't go into too much detail. Then the most caudal point that then actually continues on to become the spinal cord is the medulla oblongata. As you can see here, again, a very, very dense and complicated structure that is very highly involved in the regulation of autonomic functions. So you have centers here that are important for breathing, that are important for cardiovascular or autonomic functions of the body. So nothing that happens here is actually under our conscious control, but all that is happening here is vitally important for our survival. And maybe the most important structure here is the reticular formation that has most of these very important centers. Then the thalamus is kind of the relay station that, as Leon mentioned, all sensory information go through except for the one for smell, for the olfactory sensory information. So we have all qualities, so the quality of touch, the quality of this proprioception, though where we are, the conscious knowing of where we are in the space. Everything goes through this thalamus and then gets projected onto different parts of the cortical surface. So, for example, here the ventral anterior and the ventral lateral nucleus receive information from the cerebellum as well as the basal ganglia and also the motor cortex to then integrate this information and sending it back to the motor cortex in order to integrate and coordinate this movement. And yes, it has two different separate parts, the right and the left and the right thalamus, and each of those have over 100 nuclei, all with different functionalities. So again, a very, very highly complex structure. And then just anterior or rostral to the rostral and ventral to the thalamus is the hypothalamus, which has also a lot of different, which you can't see here, but a lot of different interconnected nuclei with different functions. And this hypothalamus is kind of responsible for a lot of autonomic functions, but is also kind of, it's not separated, but kind of functions on its own. And is very much involved in control of hormonal pathways because it's interconnected with the pituitary gland where a lot of hormones are created, and also creates some hormones itself. So for example, oxytocin or ADH and other hormones that actually regulate the release of hormones in the pituitary glands are produced in the hypothalamus. Yeah, this is also involved in the regulation of our circadian rhythm. And then the rushing through, but as I said, it's very complex structures, and yeah, I'm trying to keep it short. Then as the one of the last subcortical structures, the cerebellum, which is actually the so-called little brain, and also has sort of a structure of a brain on its own. So this has a outer cortex with gray matter, inner cortex called arbor vitae with white matter, comprises about 10%, 11% of the brain mass, and is basically its most basic function is movement coordination. It also has a lot of different functions, but this is the most important one. And for this, it receives proprioceptive information, usually from the ipsilateral side, so from the same, so the hemisphere from the same side that it lies on, and then integrates this information, going back and forth with other motor parts in the brain to coordinate in this movement. And you have three functional different parts and also structurally different parts. So they have this anterior lobe, you have a posterior lobe, you have this vermis that actually looks like a worm, sorry. And this third lobe, the lobus flocconodularis, which contains this flocculus and the nodulus. And usually on the medial part of the cerebellum, there is the control of the trunk and neck and head and neck. So the parts that are more closely to middle midline of your body, and on the lateral parts of the cerebellum, you have the control of the extremities, so hands and legs. And therefore, if you have a lesion rather on the lateral parts of the cerebellum, you have problems in coordinating your hands, your feet, something like dysdiadochokinesis, where you can't do this hand-eye, hand-nose movement, and so on. And yeah, I'll leave it at this.
Okay, and finally we come also to, because now we have discussed a lot of the structures of the cortex and what is in between, we have omitted for by now. Therefore, I shortly want to say something about the white matter. As Konstantin already said in the beginning, the white matter consists out of the axons of the neurons, so the projection fibers starting from the somas of neurons, and they are surrounded by the glia cells. And we differentiate between three types of white matter tracts. And this depends on where they start and where they are going through. So the first and the easiest form are the commissures, so commissural fibers that are going from one hemisphere to the other hemisphere, typically also to homologous regions, but also other way around. And they have to go take a passage through one of the possible connection points, because the hemispheres of the cortex are not completely connected. They're only connected via the corpus callosum, the big connection in the middle, but also by an anterior and a posterior commissure. And when we stay inside of one hemisphere, we call this association fibers. This is called that way because it's most important for abstract processing inside of the cortex, which often also takes place in association areas. But this is just a general term because this is something kind of association that takes place inside of one brain hemisphere. Therefore, this is this type of fiber. And then we have the projecting fibers. Typically also the prototypic forms are the big pyramidal cells of the motor cortex that are distending, so very long axons that are going from the hemisphere down to the efferent projections to the rest of the body. So, for example, to muscles, but also possibly, for example, to to other organs. And as you can see here, typically, if it's the pyramidal tract, then typically they cross in the pyramidal crossing and are projecting to the opposite side of the body in the end. And shortly, what have we omitted so far? These are the deep nuclei of the cortex. So inside of the white matter, we also have gray matter structures that are called nuclei, and the most well-known one are probably the basal ganglia. Because of time reasons, I think I will really shortly only mention that you all know basal ganglia are very important in movement coordination, but also movement to start movement and to initiate movements. But they are not only important for movement, they are part of three important networks. So the motor network, the limbic network, and an association network. So very complex structure consists of very complex parts. Therefore, I will not talk anymore about it, just important to mention them. And there are also other deep nuclei, for example, near the ventricles and so on, which we also will now not go more into detail, because otherwise we will not end this talk today. And therefore, as we have now given a very, very quick, and I know we rushed a bit, overview about the structural anatomy of the brains, we all know that more interesting and also more from a more modern perspective would be the functional parcellation and the functional anatomy of the brain. And we have also mentioned the functions of different structural parts, but we also know that there are approaches to understand the functional parts in a more technologically advanced way. Here, for example, you see the parcellation of Glasser et al., which Leon Martin talked a lot about in the last lecture. So therefore, we also want to enlighten a bit how we can get more information about structural parts of the anatomy of the brain, which typically do not end when there is an anatomical border as a sulcus, or just the name of the frontal lobe ends. This does not hinder neurons to build networks, functional networks that go beyond this. And just Leon, as I would just go a bit over the time, so maybe you can hurry, but I think we should finish the talk.
Yes, hi from my side. I'll hurry through the last part of the presentation. But first of all, we want to link you to the Interactive Brain Atlas from the eBrains website, because there you can explore like those areas we talked about in a three-dimensional space with a higher granularity. So there's the Jülich parcellation that is built on psychoarchitecture, and also you can explore white matter bundles in superficial or subcortical white matter tracts. And you can also explore in 3 dimensions the so-called big brain, which is usually it should zoom in, but if the video is lacking, now it's working, which is on a micrometer scale. So you can zoom in really closely and see the exact certain layers of the cortical ribbon. And yeah, there you can zoom in into the micrometer scale and take a look at how the brain looks like in this kind of resolution. So I posted the link, Leon posted the link in the chat. I was about to, but didn't click on send. There you can explore it on your own. And each area is also linked to data from the eBrains knowledge graph, which is kind of needy, I think. So back to the topic. So why do we speak about those really complex neuroanatomical terms? Because knowledge is usually represented in natural language in scientific publications, and we want to extract knowledge associated with certain brain areas. And therefore, we need a representation of an anatomical ontology. So all the terms Leon and Konstantin have said so far are defined, and then we as Konstantin said, we transferred those defined anatomical terms to a three-dimensional coordinate system of the brain, which you can see on the left-hand side, the TV base atlas with 379 areas consisting out of the 360 cluster areas and 19 subcortical areas. And there we mapped the semantics of brain anatomy, so the terms represented in language, to the 3D coordinates of the parcellation. And there we can see that with these terms defined in the Huberon ontology, which is an anatomical ontology, we have a complete representation of the brain with at least one superclass is defining each area of the parcellations. There are more subclasses of the Huberon terms, especially in the subcortical areas, because we used a rather broad parcellation of the subcortical areas, so only the hippocampus and the thalamus and the cerebellum, for example. But as you heard, there are many nuclei inside those broader defined areas which could be addressed as well. But we used an automatic segmentation approach, so kind of for neuroimaging processing. So we segmented only the broader areas. But you can see there's a quite good representation. And yeah, so this mapping between Huberon and the semantics and the 3D anatomical coordinate framework of a brain has two benefits. First of it, if you can't remember what Konstantin and Leon have said, you can type in a search engine which area you want to see, and it's represented in this 3D brain. But also you can map knowledge, like you see here, the knowledge about the Wernicke aphasia or the Broca aphasia. So we've written a script and a Python package that maps associations represented in literature to those specific areas of the three-dimensional brain. So it extracts with natural language processing information how relevant the brain areas are in the context of the search. For example, here the Wernicke aphasia, and on the right side the Broca aphasia, as you heard in the talk. And you can also see that the most relevant areas are the Wernicke area for the Wernicke aphasia and the Broca area for the Broca aphasia. And of course, other areas are relevant for this as well, because it's always a dynamic system of interconnected brain areas for that case. So it's always a system of brain areas that is relevant for a certain disease or pathology, for example. And what we've done here, we extracted, now it's repeating, but we have extracted, like if you've seen here, knowledge from over thousands of publications and extracted the association of those terms with the brain areas. And this was possible by transforming the semantic representations of brain anatomy, all the terms, into a coordinate frame. This whole software is going to be available soon with a TV-based API. So basically, it's a search engine where you can type in specific concepts of diseases, or proteins, or biological processes, and it will calculate the relevancies of the different brain areas and gives you back a map like you've seen for the Broca aphasia, where the Broca area is the most relevant for this query. And on the next slide, you can see another example maps for the term frontotemporal dementia, and you see the map is giving you relevance for the frontal lobe and the temporal lobe, and for parietal lobe, which is also seen in PET studies, really important for dementias or Alzheimer's disease. So it's extracting knowledge from literature and gives you back this maps on which brain areas are relevant for this term. Another thing what we've implemented is that it's not always data is not always available in one parcellation. So we created a framework for atlas interoperability. We do not have voxels when we speak about anatomy. We have terms like temporal lobe, frontal lobe, and all those terms Leon Konstantin told you. So we have a kind of an inherent parcellation of the knowledge we can extract from the literature, and to compare it with other parcellations or data that is available for other parcellations, we can reparcellate the data to other atlases of the human brain, which are functional or structural, or with made up with different modalities. So how does a TV base look like? We've showed you in the voxel space. So we mapped the representations of anatomy for a specific search query onto a three-dimensional coordinate space of a brain, so of a template brain. Here it's in the MNI space, and or we can represent it also on the cortical surface, on an averaged surface that is inflated. But we can also investigate the publications that are processed by the natural language processing algorithm from this Fraunhofer Institute. We've implemented, and there we can take a look at what publications are associating, for example, in this case, the hippocampus with our search query neurofibrillary tangles. And there we can go into detail and analyze the underlying foundations for this aggregation of knowledge, what we've done with our TV base maps. And you can see on the right hand side the different brain areas. Most of the terms should sound familiar right now to you. And there's a relevance measure calculated for each brain area. So it's kind of a comparison, a derivative from information theory that compares how probable it is that this term of anatomy is combined or associated with our query. And a high relevance means that there's an over proportionately high statement of this term in context of our query. Okay, so with this framework, we've mapped thousands of different genes, so the whole genome, so the association, the spatial patterns of the whole genome defined in the HCNC, the Hugo Genome Nomenclature Ontology. We mapped also all biological processes which are defined in the gene ontology, which are kind of weird name, but there are rather biological processes defined. And we mapped all terms that are listed in the medical subject headings ontology, referring to psychiatry and neurological diseases. So some of the maps we've mapped for the mesh ontology you've already seen for the Broca aphasia, Wernicke aphasia, but also for the apraxias or auditory perception. And yes, so you might think that this automatic knowledge extraction has some biases. That is true, and we've thought about how to validate this approach. And the first of all, it is, can we map brain areas itself that are not directly linked with our Huberon ontology? So we mapped brain areas defined in the mesh ontology, so with no clear link to the Huberon ontology, and compared it with with our corresponding transformation matrix. And as you can see here, that's a really high correlation. That means that no matter how the brain area is, or where the brain area is defined, we can extract the right region in the three-dimensional space of the brain. And we can extrapolate areas that are not defined in our initial Uberon transformation matrix, for example, the Broca area or the Wernicke area, with this approach. So this is a quite nice first step internal validation that our mapping process at least produces plausible maps of the brain. For the knowledge extraction, we then applied a measure of simple face validity. We mapped many functional terms of brain functioning and compared it with established knowledge reviewed in many publications. And as you can see here, in panel A, you see basic perception that is derived from clearly defined anatomical areas. For example, in the visual cortex, visual perception takes place, and you can see a kind of nice add-on that if we query the visual cortex, you clearly see only the areal borders of the visual cortex as highly relevant, and you see the systems approach here, the visual perception, which is of course in the primary visual areas mostly relevant, but is also projecting to other areas in the dorsal and the ventral stream for different kinds of perception. So the whole brain has is a system that computes the sensory input that is arriving at the primary visual cortex. So same here with auditory perception, but we can also map more complex cognitive functions like executive function, the map as you've seen before, or the theory of mind, which literature also tells us is really relevant in the temporal parietal junction, and of course the prefrontal cortex. There we can map neuropathologies. You've all seen, and what you've also seen, we can map different pathways. So the dorsal stream for pattern recognition, and the ventral stream for space perception. And you can also see how many publications are listed or are extracted and analyzed for and for those maps. So it's all from 27,000 to 900 or 200, and those are all publications that are annotated with the term we're looking for. So we have over 27K publications that are associated with the term space perception. And from those publications, we can extract the anatomical areas that are reported and then map it to the three-dimensional space. And as you can see, at least from a term of face validity, it looks legit. And as a last step, we also implemented a comparison for an external validity to see whether other coordinate-based meta-analysis frameworks if we can reproduce the results of coordinate-based meta-analysis frameworks like those of NeuroQuery, which assigns the coordinates reported in functional imaging studies, so the peak activity coordinates, to specific terms you can query. And we systematically mapped all terms available in NeuroQuery with our approach TV-based. And you can see there is a sufficient, but also not a one-to-one correlation between the maps from NeuroQuery and those from TV-based. But this is a rather methodological difference, because NeuroQuery is reporting peak activity coordinates. So it's rather defined areas in the primary or the most relevant areas with peak activities, and TV-based maps also all associations with it, so in a rather more systematic fashion. And as you can see with the term hallucinations, for sure, the most prominent hallucination is an acoustic or auditory hallucination, but there's also there are also kinds of visual and other sensory hallucinations, which are not only active in the primary sensory areas of the brain, but also are integrated and cognitively reviewed basically but with other brain areas. So it's also a network of brain function that underlies underlies an hallucination, for example. So TV map base is able to map more brain areas. Therefore, the rather low correlation in that case, but the peak activity area has an accuracy of 0.9, I would say here. Okay, so sorry for the slight delay, but last two slides. This is just a summary of the methodology, so I will be quick here, because what we've done with semantic knowledge, extracting it with natural language processing algorithms, compare it with the three-dimensional space of the brain. I've told you. I've also told you the right panel of the last slide, where we have the neuroquery, but association, and we can, with eBrains, with this interactive viewer, for example, we have in the same anatomical framework, in the same coordinate framework, we have links to empirical data. So we have a common coordinate framework that integrates semantic knowledge from coordinates, peak activity coordinates reported in the literature, and empirical data. And this is, with that framework, we can integrate all of those three modalities to create maps and to compare it with empirical data, and to reparcellate it into different parcellations, to compare it, for example, with available parcellated data in the Disikan-Kiliani or the Jülich brain parcellation, which is the main parcellation in the eBrains atlas. And for example, if you want to extract receptor density data from eBrains, this is parcellated data we can extract, and with our reparcellation method, we can then directly integrate empirical and semantic knowledge into one combined framework.